Okay, so we're going to begin chapter 9 now, and chapter 9, a lot of it's going to be review for you, especially 9.1 and 9.2, and it's about cell division. So one of the key characteristics of life, as you probably recall, is reproduction. If you want to continue on living, you need to be able to reproduce. And the way that organisms reproduce really starts with cell division. So you probably are familiar with mitosis and meiosis. That's what chapter 9 is all about. In single-celled organisms, if you divide one cell, basically you've divided and reproduced the entire organism. And so those organisms usually divide by binary fission. But cell division also allows for eukaryotes to reproduce and develop. And then once they're fully grown and reproduced, they use cell division to repair and reproduce single cells to help replace or repair them as they're needed. And so cell division is one part of a bigger process called the cell cycle. And the cell cycle is really the follow through of the life of a cell. So the main purposes of cell division are reproduction, as we talked about, growth and development, and also renewal of old or dead tissue. So 9.1, we're just going to get a little introduction to cell division. And most of the time when cells divide, they give you a completely identical genetic material in the new cell. So you'll get two new daughter cells with identical DNA. And this DNA is passed from one generation of cells to the next. And honestly, it's pretty amazing how rarely there are errors in the DNA replication process, considering how complex it is. The DNA in a cell constitutes the cell's genome. So all of that DNA makes up the genome. And in a eukaryotic cell, there are many DNA molecules, whereas in a prokaryotic cell, most of the time there's only one. And then you're also probably familiar with the fact that DNA molecules are packaged into things called chromosomes inside the cell. So the chromosomes are basically the things that hold the cell's DNA. So this would be like the nucleus of a cell, and this is the DNA. And then the DNA in this case is found in what's known as the chromatin, which is a complex of DNA and protein together. And the protein kind of helps wrap that DNA up really tightly. Each eukaryotic species has a unique number of chromosomes in each cell nucleus. So humans, for example, have 46 chromosomes, 23 from each parent. Other organisms have fewer. So, example, the fruit flies only have 4, which is why they're really easy to study. So somatic cells, which are your normal body cells, have 2 sets of chromosomes. So like I said, we have 46 in humans. That's 1 set from mom, 1 set from dad. The only cells that aren't somatic cells are referred to as gametes. These are your reproductive cells, like your sperm and your egg cells, and they only have 1 set of chromosomes, and that's because when the sperm and the egg come together, 1 set and 1 set gives you 2 sets. So that's why those only have 1 set. When cells are going to start dividing, the DNA is replicated and copied, and then the chromosomes condense into something that is actually visible. So normally they're just hanging out in the cell. It looks like spaghetti. It's chromatin. But once the DNA is replicated, the chromosomes condense, and you can actually see the individual chromosomes. They kind of start to look like worms. And each duplicated chromosome has 2 sister chromatids that are joined identical copies of the original chromosome. So the chromosome is normally by itself, but then once it gets copied, it looks like an X. And the X is held together at the center at a place called the centromere, and that is where the 2 chromatids are attached. So you can see here is an example of a chromosome. This is one sister chromatid. This is the other sister chromatid. They're duplicated. So this is after the DNA has been copied. And you can see they're held together here at the center or the centromere. Once cell division occurs, these 2 sister chromatids are going to separate and move into the 2 nuclei of the new cells. And once they're separate, now they're no longer sister chromatids, they're once again chromosomes. So you can see this would be before cell division even begins. We've got a chromosome, and then after the DNA is replicated, the DNA you can see is duplicated, and it is found in those sister chromatids. So this is exactly the same DNA, providing there were no errors, held together here at the centromere. And then after cell division, the chromosomes are going to separate and eventually end up in two separate nuclei with identical DNA in each one. And that's how we get those identical daughter cells. Eukaryotic division of cells consists of two major parts. One is mitosis, and mitosis is the division of the genetic material in the nucleus. So that's where the DNA divides and the chromosomes separate. Cytokinesis is, as you can imagine, cytocell, kinesis, the movement or division of the cytoplasm. So that's where the cells actually separate. Gametes are produced by a different version of cell division called meiosis, and we'll talk about that later in another unit, but we're going to start with mitosis. Meiosis is a little bit different because you end up with non-identical daughter cells, and those daughter cells only have one set of chromosomes, so half as many as the parent cell. So that process is a little bit different. This unit, we're focused on mitosis. And we'll stop there and come back to talk about 9.2.